Dr. Shukla said okay, so okay, genome browsers, because it is being recorded, so I will repeat. So yeah, genome browsers are important for visualizing genomes, and genome browsers are not just for visualizing, but you can use that also for downloading the data. Uh, Genomes are quite long often. For example, the human genome with over 3 billions of nucleotides, you want an efficient tool for analyzing the data, for visualizing and analyzing the data. There are many genome browser tools, but we will focus on Ensemble and UCSC genome browser. There is a genome browser at NCBI also, but it is not frequently used. Okay. Okay, so next slide. Okay, so yeah, genomic DNA is organized in chromosomes. Genome browsers display ideograms, which are pictures of chromosomes, with user selected annotation tracks that display many kinds of information. Okay, so let's actually get to next. Okay, what happened? Okay, it's going. Oh, I don't know why this thing is not working for some reason actually. So I will do from here. Not worry. Yeah, Ensemble genome browser. So actually, why not we should do here? We'll go here actually. We were doing here. So we'll go to Ensemble and then we'll click on Ensemble. Okay. Ensemble, and then we'll type beta globin or just HBB. HBB is hemoglobin subunit beta. So HBB. So HBB, actually it came pre-populated, so I will select this one. Okay, HBB, and then here it is, the hemoglobin subunit beta. Okay, and you can see here this gene actually the structure. As we discussed, there are, so these blocks are exons. So there are 3 exons, and these are variants of beta globin. Okay, that are there in this database, and I want you to download the data. So how to download the data from Ensemble? And of course, it does provide lot of information. Location, it is on chromosome 11 from this position to this position on the reverse strand of the DNA molecule. Okay. Okay. So if you want to download data, click on export data. Okay. Click on export data. And then you have all these options. So I will select FASTA sequence, which is good. And the featured strand, okay, that's good. No flanking sequences, don't want any flanking of the beta globin. And then all these options, cDNA, okay, I want to see cDNA, coding sequence, peptide sequence, 5' UTR, 3' UTR, exons and introns. All this is good. So click next. Okay, so okay, I will look HTML format. So here it is. Okay, here is the data that I got it. So if I ask you, you can get it from NCBI, you can also get it from here. Okay, and you can download this data. So you can see the first one is, so there are variants of beta globin. So it has provided you data for each of the variants. So let's look at the first one. So it starts with greater than sign, and when something is starting with greater than, it is providing you information about that sequence, the name of that sequence, and so on. Okay, so it is a sequence identifier, greater than sign. So it is telling you that this is cDNA sequence. okay, protein coding sequence. So cDNA, and cDNA, as you know, cDNA, so a messenger RNA reverse transcribed, that is cDNA. So introns are not there because messenger RNA for human genomes or eukaryotic genomes with introns are spliced out, right, and then it is reverse transcribed. So cDNA will not have the introns. Okay, so cDNA, and then you can see the cDNA sequence here. But remember, the cDNA would have UTRs. Okay, it is not that all nucleotides will code for amino acids. So then look at CDS. So CDS means coding sequence. So each of these nucleotides will be part in coding for amino acids. Okay. So, and you can see for CDS, the first codon actually is, you know, ATG is almost always used as a start codon for eukaryotes. Okay. In prokaryotes, there could be multiple start codons: ATG, CTG, TTG, GTG. Okay. So, CDS, and then next is exon. So, first exon. Okay. And see here that, okay, this is not starting with ATG, which means that a part of this is 5' mutant, and that is why cDNA is not also not. So you see ACA, here also ACA. Okay. So this is actually the first exon is starting here in the cDNA, but a part of the first exon is 5' UTR. So that is not included in CDS, because CDS will start with the start codon, which is ATG. Clear? So try to understand this data, because otherwise you will get confounded, because it's a huge amount of information contained in these databases. So this is first exon, and then is the next exon, right? This is the second exon, and then the third exon. Okay, the 3 exons are there in beta globin gene, and then there are 2 introns. So this is intron 1. And this is intron 2, okay, and then peptides, so protein sequence. So this is the protein sequence. So this is beta globin protein, okay, and then UTRs. So 3' UTR, and then 5' UTR, okay. UTR, you understand, that could be part of cDNA, but they are not going to code for amino acids. So they have to be taken out when you have the CDS. And now starts the second variant. Okay, so I will not go into this. There are many variants of beta globin, so they have provided information about all here. So you can download this data. Okay, you can copy paste or download from your browser this data. Okay, so yes.
We can use to download.
Oh, you can use to download. Oh, yeah. So I think often you will download in the text. So if you just copy paste, so it might give the option to export data. So let's see. Actually, it gives the option of that or not. So all this, and then next. So these are the HTML. Okay. You can get text. TXT, text format. So yeah, here it is actually text. You can then download this, or you can, if it's a large data, then compressed, so zipped data, and that you can unzip later on. Okay, so if you'll click on this, then this will download in your download folder. So it is downloaded in the download folder. Okay, so you can do that, and it is okay to download in zipped. Zipped or any zip, and then whenever you want to analyze, you can unzip. Okay. Yes. Oh, the variants of our interest actually. So yeah, when you have to analyze without focusing on any particular variant that you are interested in, select the first one, of course. because they are the one that is the reference sequence. So you would want to use the reference sequence, but if you are interested in particular variant, so I think there are 6 variants or so in this, then focus on that variant. Okay. Okay. So this was it. You learned about how to download data from Ensemble, and then we will go to UCSC. So UCSC genome browser. So UCSC genome browser, and let's go back to slides. Okay, so UCSC genome browser. So UCSC genome browser is used for analysis of vertebral genomic data. Okay, mainly of human and some eukaryotes. Because this is supported by a grant from National Institutes of Health that give funding only for analysis of vertebrates. That's why they have focused on vertebral data only. So we will do this actually. If you are interested in looking at the data for hemoglobin beta, so you will do similarly. So let's do it actually, because anyway I have provided you these screenshots, so that will help you. So I am going to again type HBB, hemoglobin subunit beta, and click on this, and then it should show you. Okay, why it did not? I don't know. Okay. Okay, HBB. Okay, so we will select HBB here. I don't know actually. This is really slow. Yeah, sometimes connection is slow. Sometimes it is. Here it is. So sometimes it is slow, sometimes fast, but don't worry, we will be able to. So you can see the structure as we discussed, the exons, the introns, and there is RefSeq curated data. So they are being shown in different tracks. Okay. But often you would want to download the data, so you can download from here, or you can use the tools for analysis of the data. Okay, so one of the tools is table browser. Table browser. So in the table browser, it came pre-populated with the position. So beta globin gene is located on chromosome 11 from this position start to this position, the end. Okay, so this is clade is mammalian, genome is human, assembly is HG38. So genome version HG38. Okay, and then group is genes and gene predictions, because we are interested in beta globin gene. Okay, that's why genes and gene predictions. Track is okay. All this looks good. So I want to download data. So output format will be sequence. Okay, I want to download the sequence of this gene. So sequence and then get output. So yeah, this also comes with the options plain text or compressed. So yeah, you have to download and use compressed. Yeah, don't download plain. So okay, so genomic data. So it gives option of genomic, proteomic, and mRNA. So we'll select genomic, and then okay. So these are the options. So do you want to include promoter or upstream? So then you can select. Otherwise, don't select. You want to include 5' UTR? Okay, I want to. CDS, that's fine. 3', that's fine. Introns, all that, and one faster record per gene. That's good. Exons in uppercase, everything else in lowercase, so that you can discriminate between exons and introns. So remember, exons will be shown in uppercase and then get sequence. Okay, so here it is. Okay, so this is the first variant, and the first exon is from, so exons are in uppercase. So this one is the first exon, and this will include 5' UTR. Okay, and then the letters in lowercase, that's the first intron from here to here. Okay, and then the second, okay, so yeah, the second exon is from this location to this location. So yeah, G will get included. So you understand. So I don't care. Okay, then the second intron, and then the final exon. Okay, and then you have the second variant and so on. And sometimes you just want to exclude UTRs, you want CDS exons, you don't want introns also, and all this is good. So this one. So okay, all exons, basically the cDNA. Okay, so somehow actually it got included. So the CDS, actually CDS exons should not have that ACA, actually it should be ATG. So you can see ATG. So this is CDS, and last codon is TA. So then you will say that this is CDS. So this is a signal actually that, okay, first codon almost always in eukaryote is ATG. The stop codons are TAA, TAG or TGA always. Okay, standard genetic code. Okay, so this is how you will download. Okay, and then you can do similarly for protein and mRNA. Okay, I don't have to do it. Okay, and if you want to take a look at the statistics, then click on statistics. So there are 7 item count, which means 7 variants. Okay, and a total of those item count. So to item basis 1608. So this is the length actually. And so here it is shown actually 1608. This is the length of this gene. Location is given, and so many other data here actually. So don't worry, actually just wanted to show you that. So one of the questions is actually, if you go to this and then next table browser, which we did, and this is also you will do with me during a tutorial session. We did all this. So the question is, how many ref-seq genes and their variants are on chromosome 11? Okay, so how many ref-seq genes and their variants are on chromosome 11? So you came to know that there are 7 variants. Okay, from the statistics. Oh, so 7 variants for beta globin, but this is for all actually, all genes that are there on chromosome 11. So then let's look at all chromosome 11, not just the location for beta globin gene. Okay, so the entirety of chromosome 11. So you will type CHR, which stands for chromosome 11, and then when you click on lookup, then it will give you the coordinate. So chromosome 11 is starting with the first nucleotide at position 1, and then the last nucleotide, which is at position 135,086,622. Okay, this is last position. This is entire chromosome 11, and you want to know. So the question was that how many ref-seq genes and their variants are on chromosome 11. So you have to select ref-seq. So look at this, actually track, and then change it to ref-seq. So okay, NCBI ref-seq. So select NCBI ref-seq. Okay, because the question is asking you to get the number of genes and their variants on chromosome 11, but they should be ref-seq. Okay, so I changed for that purpose, and then summary statistics. So click on summary statistics. So there are 9,930 refsic genes and their variants on chromosome 11. Okay. So these all are there in the databases. So when you use this query, it pulled everything. How many genes are there in the human genome? Any guess? How many genes are there? You said that there are over 3 billions of nucleotides, but how many genes? How many? 20, yeah, 20. I think that's a good guess. So about 20,000 genes. So when human genome was sequenced, then that time it was announced that there are over 30,000 genes. But this number has keep coming down. Now we know that it is less than 20,000 genes. Could be 19,000 or even less. But human genome is still very complex. Some of the genome, for example, some people are here from plant institute. Plant genomes could be very large, could have many more thousands of genes in them. Could have hundreds of 100,000, over 100,000 of genes. But can you let me know what makes humans so complex so far as genes are concerned? Any thought? So even though the number of genes is less than 20,000 genes, some organisms could have over 100,000 of genes, but from some perspective, human can still be more complex. So any thought on why this could be? Yes. Repetitive sequence in the human genome, but the repetitive sequence could also be in those plant genomes, repeats. So I'm just asking you to focus on genes. Focus on genes. So it less than 20,000 genes, as you said. That number could be 18,000. It keeps changing actually, because these are gene models predicted by algorithms. But people are then doing wet lab and other assays for validating these genes. So that's how actually it has kept coming down. But I think 19,000 or so, I think is still a good number actually, good estimate. So there is a process that goes on within the cell of many eukaryotes, and that process is called alternative splicing. Okay. So a gene can give rise to many proteins because of alternative splicing. And this happens because sometimes those exons can act as exons, which means coding, but sometimes they can just act as non-coding. Okay, some of those. So it's combinatorial. And because of that, a single human gene can give rise to many proteins because of alternative splicing. And that is why now we know that the human genome makes over 100,000 of RNA transcripts, could be 200,000 of RNA. So there are only 20,000 or less than 20,000 genes, but there could be over 200,000 proteins produced by these genes. That's a lot actually. So and human can be complex because of this reason, right? So 20,000 genes giving rise to 200,000 or so of proteins. Okay, so which means that one gene is giving how many? About on average about 10 proteins, right? So it's really complex, and we are still trying to understand alternative splicing. It's really a complicated process, because without this we don't know the proteins arising from these genes. So it is important to understand the gene structure as well as the alternative splicing that goes on within the genome of human. Okay, so So and for that region actually, so 9930 is also including the variants. Okay, so all those that are there in the database, because there cannot be 50% of the genes only on chromosome 11, because human genome has how many chromosomes? I'm sorry, 23. So 22 autosomes and 6 chromosomes, right? 23rd, and it's diploid, so 2 copies. Okay, so cannot have this, cannot have, and that's why this includes the variants. Okay, so that's why in the question I added variants originally. In the textbook, actually it is coming from the textbook, it just mentioned ref6 genes, but it pulled actually everything that is there. Okay, next question. Okay, we did for sequence, we did how to download the sequence. So next question, how many SNPs are encoding exons of chromosome 11? So there are many questions that will come to your mind. Some of the questions can be solved using the computer codes that are provided by UCSC browser, and some other web servers, they collaborate. So you don't have to write a program. So how many of you know here programming? You can write a computer code. Raise your hand. Okay, none? Okay, so don't worry. I'm sorry? Oh, using ChatGPT, okay. So yeah, people say that, yeah, we no longer have to code because our servant will code for us, right? The, the AI robots will code for us. You have to just give instruction and will code for. Very soon, actually, those servants will come into market. You can purchase in a few thousands of dollars, and here I think few lakhs of rupees, and they will do a lot of things for you. But they may not be allowed to take exam in lieu of you. Actually, you have to sit and take your exam. So, but I think, yeah, world is changing because all these things are happening. Okay, so let's try to solve this problem. How many SNPs are encoding exons of chromosome 11? So, SNPs, you know, what are SNPs? I'm sorry? Single nucleotide polymorphism. But what is a single nucleotide polymorphism? Yeah, go ahead. So, for example, if I compare my genome with, for example, Stravani, and so can I write actually is where this thing there is marker or something like that? Okay, good. Okay, so let's say I am just writing hypothetical. It may not be my, but hypothetical and a small portion of genome. And let's say that this is mine, A C G T A C G T. So this is mine, and Shravani's could be A C T T G C G T. So, do you see any difference between my genome and Shravani's genome? So, where is the, so which one? Which position? Third position. So, this one, and where else? This one. Okay. So, two positions, these two genomes differ, and they are called SNPs. Single nucleotide polymorphisms. So, at single nucleotide level. Okay. So, these are variations at single nucleotide level. These are called SNPs. They are important because if these changes can happen, then protein can change, right? Sometimes they can't because of the redundancy, right? So nucleotides can change, but they may code for the same amino acids, right? But they can also result in different amino acids. Look at the genetic code table. For example, if changes happen at first or second codon positions, it is possible that it may change. At third codon positions, if you look at the genetic code table, most often if those changes happen, the amino acids they code for, they do not change, right? So, but if amino acids do change, then proteins will change, right? And because of that, problems can happen. For example, there are regions that have to be conserved because that conservation is important for the function. Okay, if that is disrupted, then you may become sick. So that is the reason it is important to document all these variations in the human genome from as many individuals as could be. So many projects are going on in compiling these variations in the human genomes. You might have heard about 1,000 human genome project, now might be more because of the ease in sequencing and how cheap it has become lately. Okay, sequencing, it's really very cheap now. Okay, so the question is how many SNPs are in coding exons. So of course there are non-coding regions in human genome. Only a small fraction of the human genome codes for proteins. Could be what percentage, you know, what percentage of the human genome codes for proteins, you know? 2%, yeah, someone said 2%. So it means that 98% of the genome is not coding for proteins, but they are still important because many of those regions could be regulating these processes. Okay, they could have some functional significance which we may not know, but still there would be some regions that may not be doing anything. People just wonder actually what those are for. Okay, sometimes people say junk DNA, but I think this is something that I believe is misused because yeah, if you don't know about that, we just can't call them junk. It is just that we are not able to decipher those regions. So we will focus on those variations in coding exons because if those changes happen in non-coding regions, then maybe it may not matter most of the time unless it's regulatory region, right? But coding exons, if those changes will happen, then maybe different amino acids, if those changes happen from those codons, and then some other protein may arise. So people have tried to document this, the variations in the coding regions, because it helps us in understanding different diseases that are caused by mutations or variations in the coding regions. Okay, so you have to solve this problem, and you will do in the tutorial. Actually, I have put the same question, I believe, but I will check again. So first you have to get SNPs. So let's go back, and you have to get a SNP, and so SNPs are variations. So mammal, human, this is good. A human genome, build 38 or version 38, this is good. And there are older versions also, you can see 19, 18, 17, 16, but you should use this one 38, which is actually the default one. So 38. And then a group is actually not genes and gene predictions, because we first want to get list of all SNPs on chromosome 11. So I'm going to change this to variation. Okay, variation, and the track changed, which is okay. So this is database DB is database SNP 155, and I think this is good. And you want to focus on chromosome 11, right? So look at the question. How many SNPs are in coding exons of chromosome 11? So chromosome 11 is good, and then all this is good. So we want to get the SNPs in a certain format, and one of the most convenient or most used format is BED format, browser extensible data. So select BED format and then send this data to Galaxy, and Galaxy is a web server. So it has many programs actually there, and you can run those programs. You don't have to worry about writing any code to solve this problem. Those codes there that will help you solve this problem. Okay. So you will select on Galaxy and then send this to Galaxy. Galaxy is a web server tool that is hosted at Pennsylvania State University, I think, Penn State. UCSC is hosted by University of California at Santa Cruz. So you can send the data from UCSC to Galaxy at Penn State. And all this is good. So get output. Okay, so you can include header, and this is the default header, which I think is okay. But you can change it as you wish, but I will keep it. And then whole genome, which is okay. So whole gene, which is okay because you want to get SNPs in the genes, right? Question was SNPs in the coding exons. Here I am getting the option of SNPs in genes, and genes contain coding exons. So I am getting that option. So let's get all SNPs in genes, and don't worry about upstream, downstream. Send to Galaxy. Okay, so it is going to the Galaxy server at Penn State University in Pennsylvania. Yeah, the thing is, yeah, just have patience. Yeah, could be internet problem, internet slow. Sometimes it is slow, sometimes. Okay, this is really slow, so I will go back and try again. So let's go back and then send to. Okay, got it. Okay, got it. So it is here actually. So I think it already ran actually. So it is showing here. Okay. But it is doing again because I repeated, but doesn't matter. It is showing that it is running. Okay, it is moving, showing it is running. So, but it has already run, and it shows UCSC main on human SNP, chromosome 11. So the entirety of the chromosome. So you can click on this eye, this eye icon. Click on this, and then you will see the SNPs. So it is loading. So I think both has run, both are actually the same job. Okay. So this is different. Uh, who you want to yes?
In tutorial session,
We will
log
try.
in.
Okay, we will try something else. Yeah, let's see again. Actually, let's see display. Yeah, it is taking time, and it is also possible that the server is responding slowly. It is possible. Okay, so it is loading. It is loading. Okay, so while it is loading, actually, let's do next step. Yeah, remain focused on the question. So by doing this, you obtained SNPs in the genes of chromosome 11, right? But the question is, you have to find SNPs in coding exons of chromosome 11. So which means that you should get where these coding exons are. Okay, you already got a list of SNPs in the genes, but now you get the list of coding exons, where these coding exons are in the chromosome 11, and then you intersect these two, right? So you have SNPs in genes, and then you have coding exons. When you will intersect the common between this, then you will get the SNPs that are in the coding exons, right? So first is SNPs in genes, and your second is the coding exons. When you will intersect, then what you will get? SNPs in the coding exons, right? So the common between these two. So this is what I'm trying to do. So the next thing what we have to do is to get coding exons in chromosome 11. So go back. Actually, I don't know actually why this problem is happening, but we will try this during the tutorial session also. So you want to get data again, and that data is for the coding exons. Okay, so you will click on get data, and get data from UCSC and bring it here at Galaxy. So UCSC table browser, UCSC main table browser. Click on this, and now what you want to get? Next step, what you want to get? Coding exons. So coding exons, so your track will be genes and gene predictions, because exons are part of genes and gene predictions. So you will change from variation to genes and gene predictions, okay? And then question was, I think ref6. So yeah, it doesn't matter. It doesn't say ref6. So you can keep it as it is, or you can change. If there is option of ref6, then I will always prefer that you select ref6, because ref6 is curated data. So select ref6, I think, which is okay. And then this is good. Bed format, plain text, send to Galaxy. All this seem good. Get output. Okay, so track is, you can change it or let it be as it is. And you want to get coding exons. So do you see coding exons? Yes. So select coding exons and then send to Galaxy. Okay, let's see. Yeah, it is running. In the meanwhile, let's see actually if we are able to see it or not. Yeah, we will see later on in the afternoon. But this has run, and click on this. Here it is. Actually, it came. So the third one is actually coding exons. So you can see the coordinates of the coding exons in chromosome 11. So yeah, this is the first one. It starts at this position, ends at this position, and this is a name and so on. Okay, so all the coding exons are listed here. So you send the data to Galaxy, and then Galaxy had codes. So you asked for this list of coding exons, and together UCSC and Galaxy provided you this data. Okay. So the first is the list of SNPs in genes. The second one is the list of coding exons, both in chromosome 11. Okay. Now you have to intersect because you want to get coding exons, sorry, SNPs that are in coding exons. This was the question. So you want to intersect these two data. And there are many different tools which are available to solve these problems. So you want to intersect, you will select operate on genomic intervals. Okay. So click on operate on genomic intervals and then look for intersect. Intersect the intervals of two data sets. So click on this intersect, and you want to intersect the first one, which is the SNPs in the genes in chromosome 11. So we'll select this. So this is the first one. And then you want to intersect this with the third one, which is the list of coding exons in chromosome 11. When you will intersect means common between these two, then you will get the SNPs that are in coding exons. Okay. So run tool. So it is all good. So you are intersecting first with third, and okay, first is here, third is here, and first and second actually are the same. So intersect these two data sets for at least one base pair, and so run tool. Let's see. So this is the fourth one actually. So it is in queue. It is not running yet. Now it is running. Okay. And we expect it to give list of SNPs which are in coding exons of chromosome 11. So basically coding SNPs. Okay, still running. Okay, so some problem happened. I think it may be because of 1, 2 somehow actually they didn't do the job. So because of that actually let's delete this. Actually we will try this again in the afternoon, but check again. Okay, so okay, maybe there is some problem. So I will delete all this. I will go back actually. There is time. So we'll go back to, so we have okay, this is good. The coding exams. We'll go back to UCSC browser again. Get data. So we want to get SNP data. So we'll go to UCSC table browser. So UCSC main table browser. Click on that. And as some problem, it happens actually. Don't worry. And then we want to get a SNP. So variation. And all this is good. Mammal, human, assembly is good. Variation, SNP, this is all good. So chromosome 11, this is good. Bed, send to galaxy, get output. And then this is okay. Whole gene is okay. Send to galaxy. Yeah. Last time I think it was some job problem likely. Okay, let's see what happens actually this time. This is number 5 running. So this is running. I hope that we will be lucky this time. So okay, dead, and let's see. Okay, again problem. So yeah, this is likely some server problem. We will explore this, but this is how you have to do it. Hopefully in the afternoon we should be. If not, then at some other point during this week actually we should be able to. And I will also explore why this is happening, and sometimes it just happens. You have to repeat it, It could be that server is not doing the job properly for some reason, overloaded or something, some problem with the database, because it is not provide any error code. So it is not clear why it is not giving us the list of SNPs. But we will try this in the afternoon, and we will try to figure this out. Okay, so yeah, take a break, and think it is a break of 1 hour or so, lunch break, and then we will reassemble here. वाईफाई का पासकोड क्या है यहाँ? पासकोड हाँ बताइए. ना कर दीजिए, हाँ व्हाट्सएप कर दीजिए. ओके, इन लोगों का खाना कहाँ होगा? यहीं पे? ओ अच्छा, वहाँ पे. अच्छा, तो सब लोग वहाँ साथ जाएंगे?
सब वहाँ जाएंगे,
ओ,
तो हम लाइब्रेरी ले चलते हैं.
अच्छा ठीक है, ठीक है. और ये